of you, but we know my math skills are stellar already, right? Is somebody writing? Everybody here, did you pick A? There are 9 of you. I can count that. There are only 7 responses. Going? Yes?
I don't have it yet.
Okay.
I shouldn't be able to get it today.
So you want to write down A so that you get the credit for it.
Okay.
That's one person. All right, you're coming. All right, I'll wait for you. Go, go, go. You're good. You're fine. So while we're doing that, thank you. We've got A for glycolysis. What does glycolysis send over to the citric acid cycle? Pyruvate. What are the other products that come out of glycolysis? ATP. Does that go in here?
No.
No, where's that go?
Use the cell.
Use it in the cell. What else comes out of glycolysis? No CO2. NADH. Where's the NADH going to go? It's going to go to the ETC. If it can. If it could do respiration, it's going to go to the ETC. If it can't do respiration, where's it going to go? Fermentation. Good. All right. So we did those 10 steps of glycolysis, and they're up here. That's not really helping so much, right? That's really small up there. But fine, we've got pyruvate. I have to get pyruvate into the citric acid cycle. In the citric acid cycle, we're actually going to move in towards the membrane of the cell. So the first thing that has to happen is I can't move. How many carbons does pyruvate have?
2, 3.
3. It's a 3-carbon molecule. I can't put 3 carbons into the citric acid cycle. So the first thing that we're going to do, and this is why I wanted to do this, so the first thing we're going to do is we're going to use one single enzyme, and we're going to convert pyruvate into acetyl-CoA. This is pyruvate dehydrogenase. And this is going to do a few things. We're going to remove carbon dioxide. When I remove carbon dioxide, how many carbons are now there? 2. All right. I am going to remove NADH. So when I broke the bond and removed that one carbon, the electrons and protons from that bond get trapped into NAD. I'm going to add CoA so that I have acetyl-CoA. Does that make sense? This acetyl-CoA is going to enter in to the citric acid cycle. Questions about that? Because I'm going to pull back down the slide thing. I guess I can leave it up while we do this. Acetyl-CoA is going to bind with oxaloacetate. Now, in glycolysis, I only mentioned a few key substrates, right? I mentioned pyruvate, I mentioned phosphoenolpyruvate, I mentioned fructose, right? So there are a few key things. Oxaloacetate is one of those key things, because we're going to be talking about a lot. When oxaloacetate and acetyl-CoA join, it's using citrate synthase. We form citrate. Oxaloacetate is a 4-carbon molecule. Acetyl-CoA is a 2-carbon. How many molecules of carbon are here? 6. Does that make sense? That's where we get the name of the cycle. So the next few steps, the goal that we're going to do is we're going to be pulling off carbon dioxides. How many carbon dioxides do I take off? 2. Which means that by the time I'm here at succinyl-CoA, how many carbons are here? 4. So the halfway mark, what I've done is I've removed the 2 carbons that came in. Every time I remove a carbon, I want to trap those electrons and protons in NADPH or NADH. Those are my carrier molecules. Where are those going to go? Electron transfer chain. So at this point, I've removed all the carbon I'm going to remove, which means I've generated most of the electrons and protons. Not all of them, but most of them. The remainder of the cycle is to remake oxaloacetate and conserve as much energy as I possibly can. Does that make sense? Okay. The other big key one that we'll talk about later is alpha-ketoglutarate. Both alpha-ketoglutarate and oxaloacetate are going to be useful to the cell later, so I'm just pointing them out here. So succinyl-CoA can get transformed into succinate, and when we do this, I know it's a little hard to see, we're going to generate GTP. That can be converted to ATP. Is this oxidative phosphorylation or substrate level phosphorylation? Substrate level. This is one molecule of GTP or ATP. This is substrate level phosphorylation. Does that make sense? I'm not using a membrane. I'm not using anything big. Just generating one molecule of ATP using an enzyme. When I do that, I'm going to generate succinate. Do you remember when we mentioned this yesterday? Where was succinate dehydrogenase? Not in glycolysis.
Fermentation?
No. What's the other thing that we haven't talked about today, but in detail, but we talked about yesterday? Electron transport chain. This is complex 2. This is where FADH2 gets made and directly dropped into the electron transport chain. Does that make sense? Okay. And then I'm going to keep changing. Every time I change, if I break a bond, the electrons and the protons are going to get stored in NADH, FADH2, NADPH, and those are all going to the electron transport chain. Eventually, I'm going to recycle oxaloacetate, and I'm ready to do this again. For each molecule of glycolysis, how many times do I do this? Remember, 1 6-carbon glucose molecule gets broken into 2 3-carbon pyruvate molecules, which means this happens twice. Does that make sense? Okay. So overall, here are my sums. Now, these sums are under ideal conditions using glucose, so not every organism is going to hit these totals. But these are like the optimal conditions that you can do. In glycolysis, I put in glucose and NAD. I get out pyruvate, ATP, and NADH. Now, in reality, I'm only getting out 2 ATP. I'm going to produce 2 ATP by substrate level phosphorylation. Where is this oxidative phosphorylation occurring? Not in glycolysis. ETC. Good. This number is not exact. So for our NADH, I think it's on average 2.7 ATP are made per NADH, but some organisms can make more, like 3.1 or so. Some make less. So this is not an exact number, but it's on average. In the citric acid cycle, I make 1 molecule of GTP or ATP. I make 4 NADH or NADPH and 1 FADH2. And so this is including that acetyl-CoA formation with pyruvate dehydrogenase. In this case, again, these are averages and you multiply by 2. Overall, including electron transport activities, the optimal is 38 ATP per glucose molecule. Very rarely will you hit that 38, but that's the goal. Right? So, we get to do Elvis. We're going to take out Elvis. We're just doing part 3 right now. I know, you thought you were done. This is all shook up. This is actually one of my favorite Elvis songs, so we might have to listen to it, but we'll see. So for part 3, if you'll open up, now we're looking at a little bit more on the nutrition side. And so, yeah. So what we're looking at on the nutrition side, we've got different types of media. Remember last time we talked about complex versus defined, mono and dibasic and what have you. And we're talking about growth. And so you've got 2 tables, table 2 and table 3. And then you've got an explanation of how EPTA might be breaking down polyurethane and using it with the citric acid cycle to generate ATP. There's a reason why we tie it in now, right? So go ahead, let's get it to a few small groups. You've got some questions here. And if we can get them all so that we're all talking, we'll just do it by iClicker and not turn it in. But again, you want to be studying this for the exam. Go ahead. So go ahead, break into groups. This one's a little bit more complex. I'm going to say a definite 10 minutes on this, possibly longer if you need it, but 10 minutes hopefully will suffice. We are working on part 3 of Elvis. Good timing. That one should be somewhat easy.
In the medium one, the rich. Complex is rich.
Right, so complex is rich and then
binding is that?
ATP binding is that?
Yeah, I know what it is. But I'm saying, why did you say that transport instead of other transport? Because it says transport.
As far as the entry point and everything else.
Group translocation. That's the chemical modification. Group translocation. Maybe. Modification of substrate during transport.
Yeah, so you would need the polyframe to keep it.
What happens after?
It's phosphorylated and it doesn't leave the group. So, keep it like group. Definitely gram negative. Which gram negative bacteria very plastic finding proteins.
How are we doing on this? I told you 10 minutes. This one's hard. Hey, we're working right on what we need.
It's on the back of it.
Yeah, you have the entire case already. I'm going to pull up the roll to figure out if you need my help.
So you use this format, right? It's great. So it forms functional electron transport pathways, which would produce. I think electron transport is a pretty good one to do.
So, and that will produce this, and then usually goes in like two, right?
Yeah.
And so it's WK.
Someone's asking me the citrate transport. So it's a loss of citrate transport. So yeah, generated via substrate phosphorylation.
But it doesn't necessarily teach you so
How many ATPs are in the citric acid? And that's why there's
Oh,
You're still in. Justin,
What?
you're still in, so.
Okay, we'll find out. Figure it out tomorrow.
Just wanted to look for you.
I appreciate it. This is like a student.
We're all still 9, I'm sorry. How are we doing?
We're doing.
I see a couple of you talking to each other, a couple of you doing your telepathic thing.
Can we do a whole class thing?
I'm happy with that. That's fine. There are only 12 or so talk. I can try to interject, but part of these are that I really want you figuring it out too, but I can try to help.
Oh, the first question's pretty.
So first question, yeah.
Whatever.
First question,
Yeah.
Wait, have we got the first question? Are we good on those?
Yeah, we're good on it.
Let's see. All right. So, which one's complex? Let's see if we can get these.
Oh,
Is there a consensus here? That's what I'm trying to figure out. Go sit. Do this.
Go.
Run. These are not meant to be harmful to your brains, but to help figure things out. So the second part's hard.
Yeah.
So I think I'm waiting on 1. Is that correct?
Oh, I'm sorry.
Okay, everyone's good? Everyone's good. So everybody said medium 1. Why did she say that medium 1 was complex?
Because it has specific ingredients, or they have like it lists like the amount of grams and everything that you need to put in it.
Well, they both do.
Yeah, they both do. That's
Oh,
not
It allows more, like, yeast is a more complex molecule. It's
Oh,
more
okay,
like,
yeah, that is.
that one.
So yeah, you're not getting the distinct breakdown there, right? That you get in the other one, like how many grams of each type of buffer and all of that, right? So that's more complex. I don't know exactly what's in the yeast. I don't know what proteins that yeast had. Okay, so let's try another one. Which one is minimal?
Why does this thing keep turning off?
Batteries usually.
No, it's not.
Oh, then I have no idea. I'm out. I tap.
That's what it wants to use.
That's technology in general for me.
I like the one better.
Well, baby.
There's like.
Okay, so I'm waiting on 1.
Obviously, you have to.
So there's actually not a consensus here. Just so you guys know. Going once, going twice. Everyone happy with their answer?
B.
Once, twice, sold. Okay. Okay, so basically what we're looking at here, and so if you're writing, we just don't change our answer once we go over it. The answer here is which?
B.
B. So the majority got it, about 80% of the class got it. Why is it considered minimal?
Because it's more specific and more defined, like.
That is true of it, but that is not why it's considered minimal.
It's because like the E. coli won't grow in it.
That is a true statement as well, but that's not why it's considered minimal. And that's why we do these. It's really to kind of brainstorm and figure out what we're doing.
Compared to the other one, this is more recipe like that.
So that would make it defined.
Oh,
Again, correct, but
yeah,
not.
that's it. No?
So minimal, kind of. The concept with minimal media, what are we trying to get Eptum to grow on?
You're trying to get it on the polyurethane.
Polyurethane. And so what I did, or what they did, is they removed all the other carbon sources from the media. It is minimal. It only has our weird polyurethane in it. And because it only has that, it's called a minimal media. Does that make sense? So it's a way to say, yeah, it's minimal, it doesn't have so much stuff in it, it's very defined. It is defined as well. But it's minimal because you only have that one carbon source. Sometimes you could do minimal media for carbon, you might do it for nitrogen, you might do it for sulfur, what have you, but it's one source. You either use it or you don't, and you die. Does that make sense? Okay. So we've done the first part, and that's kind of the easy question, right? Now we move into question 2. Do you guys have thoughts on question 2, or do you need to talk as a group?
I don't think it would hurt to talk as you.
Okay, why don't you guys talk as a group?
Because it breaks it down.
Yeah.
Oh, it's gonna break it.
So they're really trying to figure out.
It's gonna break it down into smaller molecules. Wouldn't it break it down? Not now.
It says the enzyme degrades into citric acid.
Start with the first part. That's the big thing. Why is it important that it secretes polyuretane?
What was your explanation?
Isn't that breaking it down? Like,
That's what I figured, since it's like, it says it's big, so it obviously has to be something.
someone's gotta break it down.
Yeah.
I'm waiting until you guys tell me that you're good and we're moving. I'm not gonna push you on this because I need you to figure this out. I love this case study because it goes through everything we've covered so far.
How can a material cover atoms?
So these are our short answers you're giving?
They're not this, but it's based on this concept. I'm gonna show you, so you don't have the real part 4. I'm gonna do a part 4 with you, which I hope will help prepare you for the exam. I'm not leaving you out in left field, I promise. I'm gonna give you some guidance here. You're about to cry.
Yeah.
Yeah, no, there's not gonna be crying. There's no crying in microbiology. I refuse to allow crying in microbiology, but hopefully with my hints it'll help.
We could tie it in.
How's it gonna find its way in?
Kind of, but no, yeah. Keep going.
Okay, so if it's breaking it down, then what's the entry point? That's what I was trying to figure out.
Wouldn't it be the ABC cassette or ABC transport system? Because it expends energy to bring it in.
Okay,
That makes sense?
yes.
Because it uses up 4 ATPs to bring whatever it needs inside to make more ATP.
Okay, and then after that, it says that,
What happens after it's entry into the metabolic
Yeah.
pathway?
What happens after its entry? It goes through, doesn't it go through the citric acid cycle? Is that
It's
what-
already citric.
Yeah.
So then it goes to
So it's only
well, it already says it contains the ABC transport system.
So that's what's bringing in the
Yeah,
polyurethane
Exactly.
stuff to break it down,
so,
to make more ATP, to get energy, to do more. Well, the ABC transport system
is the entry point.
is the entry point. Is it, huh? What happens after the entry, or we go through the citric acid cycle? What is it?
That's what it's doing. It's kind of what we just went over. So.
Guys, I hear like 4 people talking. I want the rest to be talking. I don't want to have to collect this. I want you guys talking it out. Do you need my help?
Yes.
Okay. So if we are looking at question 2, given that it's a big polymer, like huge, why do you think that this enzyme is secreted?
Because we want to break it down to smaller things.
Why?
But we can utilize the smaller stuff easier than we can.
That is a true statement, but that's not the most correct statement in this case.
Because of the ABC transport system, we need to break it down into, so like bring in citrate, and that enzyme will break it down into citrate. You can take it in that way.
So I can't physically bring it into the body. You guys, you had to take animal bio probably to be here.
Nope.
Nope, never mind. Animal bio, do you know how a spider eats? Spider bites you.
Puts enzymes and breaks
It secretes its enzymes and then sucks it up like through a straw. It's kind of what the bacteria is doing here. It's releasing its enzyme and then it's just sucking up the citrate through the ABC transporter. Does that make sense? Okay, so if it is pulling it in that way, what is the entry point for metabolism then?
ABC transport.
Well, that's the entry into the cell, but where are we going in? Along the pathway of glycolysis, citric acid cycle, are we coming in pyruvate, are we coming in acetate, where are we coming in? Citrate.
Citrate, so you're going straight into citric acid.
So what have I lost?
Glycolysis.
I've lost anything I would have made in glycolysis. What else have I lost?
Pyruvate.
The pyruvate dehydrogenase molecules. What I'm trying to show you with this is that depending on where you drop in, you make different amounts of energy. So you know how we always talk about glucose? How many of you have eaten pure glucose? Right? So I actually did mouth pipette glycerol back before my bosses, told me not to do it. And I was working in the government lab at the VA hospital at the time, and they told me to mouth pipette it. Glycerol is disgusting. You don't want to eat this stuff. Like it is. My boss was an MD, PhD, and she still told me to mouth pipette glycerol. So I've never really forgiven her for that. I don't care, it's less of you. It was like 20-something years ago, but it hurts, right? What you eat gives you different amounts of energy. In this case, what they're eating is giving them a different amount of energy because it's coming in a different place. Now, you don't have to expend any of the energy for glycolysis, that's good. But you don't make any of the energy that you would have gotten in that preliminary tally. Does that make sense? Okay. So taking that now, we've got 4 sections in question 3, right? So the very first question to figure out, is there a net gain or loss of energy when we're transporting citrate?
There's a lot. You have to assume 4. To bring it in the top.
Yeah, but then
How are we doing? Oh.
Yeah.
There is a question on the board. I think there's something in the air today. My kids were like space cadets this morning trying to get them out of the house. My husband was like a space cadet trying to get them out of the house. Like all 3 of them. It's like,
there is an eclipse or something.
is there? There's got to be something.
No, I know.
Is it like full moon, eclipse, something?
So it's
One of
not
my
us.
good college
It's
friends
not
is the
us.
head of the ER at Grady. So full moon, his Facebook posts just get awesome.
People act crazy.
no, they don't.
Yes, they do.
I think it's an excuse, but it is entertaining.
I did a huge, like I'm commuting an hour, like this huge truck fell
Just-
over, so I had to stay for 30 minutes in traffic.
It's not
That was
pleasant.
like
Yeah, no.
a huge truck, I just,
Yeah, no. Gotta love it, right?
wow.
That was today?
Yeah, right now.
God, I hope everyone's okay.
I left earlier than yesterday and came
Later.
later than.
I don't know why you're surprised. Atlanta traffic.
They're saying 85 is supposed to open this week.
Yeah, we'll see how that
At the
goes.
latest time.
I'm a MARTA rider. I don't really care. I just want everybody off MARTA.
Oh.
I don't have my seat anymore. Like,
You realize some of these people are gonna know, be like, Marta was great.
they need to get off MARTA. It's too crowded for my blood. Every time I take a personality test, I am one of the biggest introverts you will ever meet. I do very well in social situations, but I relax. Quiet time, don't come near me. You may sit next to me, I don't care. I'm not that type of person, but just shh, don't talk. I'm gonna zone out on Marta. Okay, so do we have an answer here? I only have 8 of you responding.
I'm waiting on every reading.
One more?
I mean, I consistently say it's B. Because we're expending 4 to bring it into the south. That's not gaining anything.
So this is a really good word choice. We're leading up to the first exam, right? So like I'm trying to like train you to read the questions. Is there a net gain or loss of energy specifically during transport? I'm not talking about metabolism. I'm just talking about that ABC transporter coming in. All right? Going once, going twice. Sold. 90% of you that responded got it. This is a net loss of how many? 4. Okay, so we have a net loss of 4. Does that make sense? So I think the next question asked, does glycolysis and substrate level phosphorylation help balance that out?
That's gonna cause.
Okay, so first of all, why would you be like, why are you even asking that question?
Because we missed out on.
Yeah, we totally missed out on glycolysis. We jumped from taking this huge polyurethane all the way into citrate. So there's no glycolysis, there's no pyruvate dehydrogenase, there's no acetyl-CoA, we jump right in. If we had been able to do glycolysis and substrate level phosphorylation, would we have made enough ATP to pull this in?
Yes.
Yes. No,
Yeah,
because we would have only produced 2.
correct.
We would have.
So is substrate level phosphorylation sufficient to enable this?
No.
No. So what do they need on top of it? And that's where this next question, I think, see? They need, well, what about the electron transport chain? What are they doing? This is C. How can we generate a net positive? We can go through the electron transport chain, but specifically with what process? Oxidative phosphorylation, exactly. Okay, so now they want to explain how oxygen plays into this picture. So what are the key players in electron transport chain and oxidative phosphorylation? Okay, not NAD, that's a product. NADH and FADH2 are coming in, and what are they donating to the electron transport chain? Electrons and protons. Which one goes through the membrane? Electrons. Which one gets pumped out? Protons. How does the proton gradient dissipate and come back into the cell? ATP synthase, the proton motive force, the protons flow through the ATP synthase, crank the rotor, and come back through. Who do they want to meet up with when they get on the inside? Oxygen. And what do they form? There you go. Does that make sense?
Yes.
Okay. So I know, Aptem is degrading some Elvis impersonator costume. It's a little far-fetched, but do you see how you could use this type of information to walk through literally everything from microscopy and cells to basic media to glycolysis and TCA? Yeah? Okay. So I'm not going to leave you hanging on this. Towards the end of this section, towards the end of this section of the PowerPoint, we're going to do a part 4 to give you an idea for the short answer on the exam. Deal? But before we do that, we have to talk about how cool metabolism is. How many of you have heard the phrase that enzymes are rusty gates? Nope. You took biochemistry, right?
Nope.
You took cell and molecular. That's a requirement. Maybe, maybe not. For the most part, unless you're coming in a different type of path, you took one of those. Enzymes, have these active sites. They take in substrates. They convert them to products, right? That's their job. Just the same way you saw in the video for the ATPase, though, you can reverse it. And so that's the concept of a rusty gate. If you think about, not right now, Georgia, because it's blue skies, it's gorgeous out. I assume it's warm, maybe. I haven't been outside.
It's like beach
We just do
weather.
5 hours of straight lecture for me, so I don't see the outside.
Missing out. It's perfect.
You're really not helping.
It's like beach weather. Just want a beach weather.
I want the beach. I'm like planning the beach trip. My kids are like, Mommy, when do we go? When we're all out of school. First we have to get money and do Cub Scout camp, and then we go to the beach. Anyway, so if you think about if you have a gate, a metal gate that's open, and you always go out from that gate, and you keep that gate open in one direction. If it rains and then dries out and rains and then dries out, it becomes kind of rusted open in that one direction. If I decide I want to close it and open it in the opposite direction, what am I going to do? WD-40 helps. A little bit of elbow grease, right? I'm going to push that thing as hard as I can, but I can make it go in the other direction. It's the same with enzymes. If I alter pH, if I alter temperature, you can make the enzymes work in the opposite direction. Now, some enzymes you would have to force. But like ATPase, some of them will switch depending on the needs of the cell. So in this case, if I think about glycolysis, those 10 enzymes, and citric acid cycle, those 8 plus the pyruvate dehydrogenase, if they're all going in what we would describe as the forward direction, the direction we've been talking about, what is their goal? What is the goal of glycolysis and TCA?
Or ATP.
Make ATP, that's my goal. Yeah? If the cell is running out of basic nutrients, you need to stop and say, okay, forget this whole energy thing. make some nitrogen so that I can make more protein, so that I can make more enzymes and I can do more metabolism. And so you'll stop. And so you actually have where you can reverse most of our metabolic pathways in order to do what's known as biosynthesis. So instead of catabolism to make energy, we're now going to reverse into anabolism to do biosynthesis. And so there are a few key places to do this, and I tried to mention some of them as we went along. Alpha-ketoglutarate and oxaloacetate are two key sensors to the amount of nitrogen in the cell. If you have low levels of amino acids, low levels of nitrogen in the cell, you actually will pull off alpha-ketoglutarate and oxaloacetate from the citric acid cycle and use them to make all of the different types of amino acids that you would need. Once you have enough amino acids, you remake oxaloacetate and alpha-ketoglutarate and you reverse back the opposite way. So they have a dual role in metabolism and biosynthesis, and I'm going to give you some examples of these pathways or cycles that are used for it. Your book goes into a lot more detail though. You only need to know the level of detail that we're going to discuss in class. Yeah? Okay. So this is just one example. This is the glyoxylate pathway. If you are growing cells on acetate as their carbon source, acetate has how many carbons? No?
Two?
Two. Because that's the precursor for acetyl-CoA. Is that a rich carbon source compared to something like glucose?
Nope.
No. So do they like it? No. So if I'm growing them on acetate though, I can go through what's known as the glyoxylate pathway. And so acetate we can convert to acetyl-CoA to citrate, and I'm going to jump into this pathway, right? I can pull off glyoxylate and make malate and go backwards to make oxaloacetate. So this would kind of allow me to continue this cycle. Or I can pull off to succinate and I can start doing biosynthesis for whatever I need. So the cell can go in opposite directions. Glyoxylate is going to help permit biosynthesis, but also help remake oxaloacetate to continue cellular respiration. Do you see that? So other examples of this that we'll go over briefly are the pentose phosphate pathway and gluconeogenesis. In both cases, we're trying to make sugars. In the pentose phosphate pathway, we're trying to make a pentose sugar from a hexose sugar. What does that mean in English?
5 sugars, 6
5 sugars from a 6 sugar. Now we use 6 sugar in metabolism and catabolism, right? When do we use 5 carbon sugars? Pentoses? What macromolecule has a 5 carbon sugar?
Fructose?
Well, fructose does, that's a carbon, but what class of macromolecules do you think I'm trying to make? It's on the slide.
Glycogen?
Ribonucleotides, deoxyribonucleotides, so DNA and RNA. So the pentose phosphate pathway, when do we see glucose 6-phosphate? We've seen it a couple of times.
It was in...
It's the first step in glycolysis. This is also that phosphotransferase system, remember the hot potato system, to pull in glucose. If I have glucose 6-phosphate and I dump that into the pentose phosphate pathway, instead of going to make energy, I can take this 5 carbon sugar and turn it into either ribose or deoxyribose to make my nucleotides. That's a form of anabolism. Gluconeogenesis, when you're growing cells on other types of carbon sources, they can go backwards and make 6 carbon glucose. I'm going to show you that in a moment. And they're going to use this typically for cell wall synthesis and cell material synthesis. So if I have, in this case, this is showing you for pentose phosphate pathway just in more detail, but I can take glucose 6-phosphate and I can convert it to a 5 carbon. If I'm growing them on 5 carbon sugars and I need to make cell wall material, I can do gluconeogenesis by just reconfiguring my numbers of carbons. And I'm not teaching you all of the enzymes because I don't think that we need to, especially in a 3-week course. But I need you to understand that these are 2 ways to manipulate glucose metabolism in order to make either cell wall material or nucleotides. Does that make sense? Yes, ma'am.
So you basically just want us to know that you can either make nucleotides or it can end up in gluconeogenesis?
I want you to understand the difference between the goals of these. One is if you have a 6 carbon sugar, You can make 5 carbon sugars to make nucleotides. The other is if you don't have a 6 carbon sugar, you can combine all of the carbons that you're consuming in metabolism and use it to reconfigure the number of carbons to make glucose or other cell wall materials. Does that make sense? They're kind of opposites in purpose.
Sorry.
Please don't apologize.
Do you also try to make the 5 carbon chains on the second one too?
No, not really. I mean, you can if you're reconfiguring, but typically you're not trying to. Typically, this is when you're not growing on a 6 or an even number, and you're trying to make that. Okay? So that's nucleotides and sugars. I mentioned amino acids. Who do I use for that? I'm not sneezing or coughing. That's not an improvement.
I can't control these things.
No, none of us can. What are the 2 carbon molecules that I said, or carbon compounds that I said, that would help us to make amino acids? They both come from the citric acid cycle.
The alpha-ketoglutarate.
Alpha-ketoglutarate, oxaloacetate. Good. So if I am looking for those, I can use those from the citric acid cycle to make any of my glutamate or aspartate family amino acids. They're really good at it. And so we would actually see there are different enzymes. So for instance, if I have alpha-ketoglutarate, I use glutamate dehydrogenase, and I can convert that into glutamate. Once I have glutamate, I can use that with different enzymes, so either glutamine synthetase or transaminase, and I'm making different amino acids along the way. If I need to make from the alanine, serine, or the aromatics, you're actually pulling off either pyruvate or 3-phosphoglycerate. That's the midpoint of glycolysis to make those 2 families. Or this is phosphoenolpyruvate. Charismate you would get by combining phosphoenolpyruvate in the pentose phosphate pathway, and that's how you would make your aromatics. So again, I'm not going to say to you on the exam, how do you make arginine? How do you make tryptophan? But I want you to see that by using the glycolysis, the citric acid, and the pentose phosphate pathways, if I play with them, I can basically make nucleotides, sugars, and I can make amino acids. Does that make sense? What's the macromolecule that I haven't mentioned? Lipids. So I have one more slide on this step, and then we have to figure out lipids. What? Lipids are nuts. There's a reason we don't talk very much about lipid biosynthesis, and I'll explain in a moment. For nucleotides, we talked about how you make the sugars, but not the actual nitrogenous bases. So the nitrogenous bases, you actually have a couple of different skeletons. You have 2 key intermediates, one for purines, which is known as inosinic acid, and one for pyrimidines, which is uridyl. And so these are the basics that you would then use to apply and make any of the nucleotides. But again, first I have to make those 5 carbon sugars in the pentose phosphate pathway. That's a requirement. Lipid biosynthesis is nuts. Literally nuts. So I've already confessed that I was not the world's best student as an undergrad. In grad school, though, I kind of rocked it. I started my master's, and I had a 4.0 the entire master's program. And so my husband was treating me, and he's like, here, have a piece of jewelry. Thanks. Then I got into a PhD program. Of course, it was like 10 years later. But I started my PhD program with all straight A's. Like, yeah, I know, right? Never happened in undergrad. Never happened. One semester.
It's not very common.
Yeah, it's not common at all.
I got straight A's 1 semester too.
Yeah, and then, actually mine was one of my last ones. Yeah, we don't talk about it. Okay, so my last class in my PhD, very last class, had straight A's from master's through PhD. It was biochem 2, and I love biochemistry. Until he got to this, and we had to draw each structure and intermediate and product through each of these. And I said, dude, that's not okay. And my husband said, look, I'll buy you diamonds. Do you want diamonds? I don't want diamonds. No, it's not. I don't want diamonds. I'm not doing it. Like, no. You have one test left. I don't care. So I lost it in that. I had an A- or something in that class, and he would not get me jewelry. But I don't like drawing these intermediates. That's, you make choices in life, and I made a choice. No jewelry. I was very firm on that one. He got off cheap, right? Okay, anyway. He has to deal with me, so that's his, bearing in life. So when I'm trying to make these, this first part, the key idea is anytime I want to add carbons, I'm going to add them in twos, and my precursor are going to be these acetyl molecules. And I'm always going to use an acyl carrier protein to help pull them in and add them to a growing chain of fatty acids. All right? And so literally what you are doing here, anytime you break a bond, I'm removing carbons. I'm going to try to generate energy if I'm doing catabolism. In this case, I'm doing anabolism. So I'm putting in energy to form the bond in between the new 2-carbon molecule and whatever I'm adding it to. So I'm going to keep cranking this unit. You're adding acetyl units by adding acyl carrier protein units, and it's just adding 2 at a time. That is for a fatty acid. To make it a lipid, I would add glycerol. To make it a phospholipid, I'd also add phosphate. If I were trying to make an oil or something else, I would add different constituents, because remember, the key with lipids, they don't have a single type of monomer. It's not like they're all amino acids or they're all nucleotides. They have varied monomers depending on the properties of the fat. Does that make sense?
Mm-hmm.
Okay. And that's all we're going to talk about with lipids. But we get to talk about a more fun topic, in my opinion. This is nitrogen fixation. What do I mean by nitrogen fixation?
I have a quick question.
Oh, sure.
Did you want us to memorize the diagram on the lipids?
No. I want you to go through what I said. Use acyl carrier proteins to add 2 carbons at a time for a growing fatty acid chain. Then glycerol to make a lipid. That's literally it. I don't believe in memorizing because you're just going to forget it. And we all have phones and laptops and iPads, and you can always look it up.
I wouldn't learn it if I just memorized it.
Yeah, nobody learns it when you just memorize. This is why they've actually done a shift now in medical and nursing programs. Because if you look back 15, 20 years ago, it was all memorization. You just memorize one thing after another. And then when you go to apply it, it can become very difficult. And so nowadays, if you look, the newer people coming out of nursing and medical programs are actually being forced to do group work, forced to do group assignments, analysis. It's very different now, which is also why we do a lot of group work. We're told to encourage that in STEM classes. It's a whole new approach to learning because you don't learn if you memorize. I want you to understand the purpose. I want you to understand the idea. But you don't need to draw the intermediates. Why would you draw the intermediates? Nobody cares about the intermediates unless you're a biochemist, and that happens to be your research, but we won't go there anymore. How's barbarians? Are we good?
Yeah.
I'm not that mean, by the way. So nitrogen fixation, what do I mean by nitrogen fixation? Who knows, right? It's just a word. There's nitrogen in it. Okay, what kind of nitrogen is in your atmosphere? Hopefully you're not, taking it and doing something with it.
Gas, please.
Hmm?
Go ahead.
Well, it's dinitrogen, it's gas. Yeah, and 2, can you do anything with that?
Breathe.
You probably don't want to play too much with it. Bacteria, though, can do something really cool. Not all of them. Eukaryotes can't do this. Plants can't do this. Fungi can't do this. Most prokaryotes can't do this. But there's a small select group. We're going to call them diazotropes. And what they can do is awesome. They take this dinitrogen gas that the rest of us look at like, I got nothing. They convert it to ammonia. They convert inorganic nitrogen into organic nitrogen for the rest of us to use. For all of your nitrogenous bases, your amino acids, your proteins, your DNA, your RNA, thank them, because that's the only way you get it. So this is a very important process. This is not the only time we're going to talk about it. This is the introduction to it. All right. So we have organisms that are free-living and aerobic that fix nitrogen. We have organisms that are free-living and anaerobic and fix nitrogen, and we have organisms that are symbiotic. A lot of them are going to be with leguminous plants, which is why they have a very high nitrogen content, but some will be with non-leguminous plants as well. And so this is energy intensive. I'm going to take N2 and I'm going to reduce it all the way to NH3. That's a lot of electrons. It's 8 electrons. That's huge. It's done by an enzyme known as nitrogenase, and it has 2 subunits. It has a dinitrogenase and a dinitrogenase reductase. The reduction is actually going to occur on the dinitrogenase. Remember we talked about these trace elements that we would need? There's an iron molybdenum core inside the dinitrogenase, and that's going to hold my dinitrogen. My reductase is going to keep pounding that with electrons and protons until we get to the NH3. Does that make sense? All right, here's the caveat. The enzyme is completely inactivated by oxygen. Bless you. We completely kill this enzyme if it is exposed to oxygen. That's bad. And some of them are free-living aerobes. We have a problem here. So, organisms will come up with weird ways to protect their dinitrogenase. And so as we go through, we're going to talk about different ways in the semester, all 2.5 remaining weeks of it, for how we're going to do this. But one great example are cyanobacteria. So remember we saw a picture of these when we looked at fluorescent microscopy. They've got these really cool colors. They're awesome. They help us with photosynthesis. These are all the same. They're living in a chain together. Do you see that one of them doesn't look quite like the others?
The big old bright green.
The big bright green thing, yeah. So remember, these are the ones that will go up and down in the water column using their gas vesicles. When they go up in the water column, all the little red dudes, we need them because they are doing photosynthesis. But when they do photosynthesis, what gas do they release? Oxygen. So this dude, my little green dude, does not do photosynthesis. This is a differentiated cyanobacteria. This is known as a heterocyst. As a heterocyst, it has no ability to make oxygen. It can make a little bit of energy, a little bit of sugar using photosynthesis, but it cannot make oxygen. The red cells have to help feed it with extra energy and extra sugar. But it makes the fixed nitrogen, the ammonia, for all of its neighbors and shares it with them.
Does that also go up to the surface?
The whole chain will go up to the surface, but there's no oxygen production in that cell. So it's completely protecting its dinitrogenase. Does that make sense? So if we look at the reaction, now in this case I am going to walk you through it. We're going to talk about this reaction probably another 5 or 6 times in the next 2.5 weeks. We don't have to memorize numbers of electrons and all of that, but I want you to understand the pathway. So I have to generate electrons. The pyruvate that we have during glycolysis, or actually at the end, donates electrons to the flavotoxin. That in turn will reduce the dinitrogenase reductase, and that, remember, is going to provide the electrons and the protons to the dinitrogenase to enable the reduction. Does that make sense? I get electrons from pyruvate. The electrons go from pyruvate to flavotoxin, from flavotoxin to dinitrogenase reductase, from dinitrogenase reductase to the N2 in the dinitrogenase to make ammonia. You can kind of get rid of the rest of it and just know the flow. Is that a deal? Okay. So let's talk about how we could use Elvis to figure out all of this, right? So this is the type of thing that you might see on a test. Now on a test, and I'm going to be completely honest, I haven't made your test for this week yet because I'm human and I'm doing my best to get through it, right, with you. You at least have next week's notes. At least I'm ahead on that. But this is the type of thing that you might see. So the short answer question might have things about types of media or what type of microscopy would you use and why. So there are going to be some basic, what I would consider almost like low-level taxonomy questions, right? I think they call that like low-hanging fruit. Then you would see stuff like this, where this is a little bit more advanced. And so if the short answer question were, let's say, worth 20 points, this part might be worth anywhere from 4 to 6. And so you might see a story. In this case, the story is DuPont has now tasked you with creating a new generation of Elvis coating to protect against future biometabolism. In other words, we're going to do this again. I don't want this to come back gooey and messy. How would you approach design? What would you do to protect this material? Are there structural aspects for the polyurethane or whatever you're going to use? What other types of material might you consider? So just take about 5 minutes and brainstorm with the people around you. How could you answer a question like this for an exam? That means talk.
Yeah. With the but no, you're sitting in the paper, but you can make it up in different materials.
I guess just do a thorough analysis of every single thing and then put it in the type of material.
You usually don't know. That's how you, if you can get something that can go up into the, because the reason they use a lot of the urethane is because it's a look. But then they realize that also is degraded. So if you use something that gives it similar look, but doesn't degrade.
That'd be fine, but do you think they care after? Maybe probably, like there's a whole thing.
They want him to look like Elvis. But
Yeah,
it's
we
like
have to try to look like Elvis. That's the goal.
seeing the material that doesn't get degraded by
What types of material would you consider?
rubber.
Like structural
Maybe. I don't know what makes up polyurethane.
Maybe like some type of bubble.
I'm not asking you to be a chemist. That's
Or
not
what
the goal.
You can use polyurethane to make.
You could make it like
Structural.
like a vacuum with like no oxygen in it. I don't know.
So do we have some ideas? Okay, what are our ideas? Give me 3. 3 different ideas, not one person, but 3
That's a
ideas.
terrible idea. I think I don't know how you would do that.
That's okay. Share
You could
it.
use rubber.
Rubber?
'Cause
Okay.
Rubber didn't degrade when the boss put it in the.
Okay, so rubber didn't work, so we're going to put rubber. We're going to have a rubber Elvis. That would make it interesting. I don't think I'd want a rubber suit, but if you can mold the rubber appropriately, you can change the material. Bless you. Didn't you say one and done?
I know.
You did, didn't you? All right, go on. Give me a second idea. So I've got rubber.
You can try to experiment with different types of, like, different materials that you don't know yet and just see how they react with the lycra.
Okay, so I'm going to see, okay, so you're combining different materials with the lycra?
Well, I want to test it first and then.
Okay, so maybe there's something that would act as a preventive mechanism. Okay. Guys have exhausted the structural aspects. You got to consider other things maybe, or,
Something that degrades aptum.
oh, maybe put something in that would eat ectom, a predatory bacteria. Okay, I said 3, but it was the same 2 people giving me all 3, and they talked together. Another group must share an idea. Going once, or you have to turn it in writing. Someone share.
Someone just share something.
It could be a crazy idea.
I don't know. It only works in oxygen, so if we could find some type of way to have like a vacuum seal around it somehow.
Yeah, if this is an aerobic process and they need oxidative phosphorylation to do this, put him up, put our probe up. If all we're doing maybe is taking pictures, if we're not actually taking soil samples, you can put an anaerobic chamber around it, and that's totally fine.
I was thinking capsules or slime or something like that.
Capsules or slime, so something to prevent metabolic, eating of it or even just sensing it. So what some people have talked about in the past is putting in something that would confuse their communication. Remember they were doing chemotaxis? So if you put in something to confuse the chemotaxis. Another idea would be to add a different food source that was easier for them to consume. Remember, it's an energy loss to pull in the citrate. Glucose, just coat it with sugar. What?
Can we change the pH or the temperature or something so that
Sure.
they go into the endospore?
Yeah, if they are sporulating, and I don't think they had dipiclinic acid, but they also didn't test for it. So maybe they're sporulating and you can change it so that they become spores. So there's a lot that you can do. And the reason to bring this up now is because A, I'm trying to give you ideas for an exam. But I also want you to relax on the exam. When it comes to this type of question on the exam, there's not one answer. Crazy ideas are not always wrong. I want you to take a risk. If you have a question, you can say, this is what I'm thinking. Is this like totally crazy? I'll try to link you up. If you are jumping the gun and doing some genetic engineering to invent an alien microbe, I might say, I think you're outside of the scope of this question. Cool, but outside the scope. You can ask that, but I want you to realize that any of those questions with proper explanation, like you can't just say, I'm going to put them in a vacuum. Why? Tell me why. You did with the oxygen versus no oxygen. Why are we going to use rubber?
Because rubber didn't get degraded when you
Right.
sealed
You'd
it.
have to just say that on the exam. Does that make sense? So there's going to be the quote verbatim, define this part of the short answer, and then there'll be the now what? And so you'll have to do both, right? And there's a variability in the now what. Does that make sense? Okay. So what I'd like to do then, we are going to take a break in about 10 minutes, but I'd like to keep going, and here's my thought while I'm talking about this. I'll pull up this video. My thought is that I would like to get as far as possible. That's not a download, that's a video, you goofy. So if I can get far enough, my thought is that we can then have a little bit of time to talk about it, right? That's kind of a good thing to do. Does that make sense?
Yes.
Okay, let me see if I can get you this video. If I'd like to. We'll see. Here we go. If I can copy it, it should go to it. This is our Stanley Falco, because he's funny. He's not ducking this time.
The whole issue for a microorganism is replication. Every microbe sitting in its mother's flagellum learns that the first thing that it must learn to do is to replicate. And so when you watch microbes replicate, they do so by simple binary fission. And you can see that you simply become surrounded by them over a period of time. Now, watching this and knowing that this doubling occurs every 20 minutes, and if it went on freely, a single microorganism would literally grow to something that was about 23 times the volume of the Earth if it actually was permitted to go unfettered. That, of course, doesn't happen. But it gives you some idea of why a surgeon fears even a single microorganism falling into a surgical field, and why your mother told you to always clean out a wound. You don't want these dangerous kinds of things in you. But this is what microorganisms do. They have to replicate. So the whole thing from entry and all the steps that it has to go through has this one function in mind: replication. Make more of yourself. And the reason that organisms to make more of themselves is either to persist or it's to go to a new host.
All right. So I wanted you to get an idea of what we were looking at before we start talking about it. Now, what you are seeing is the simple idea of binary fission. And if you watch behind Dr. Falco, you see the bacteria elongate, split, and separate. And that's what you were taught when you were taught cell division. We usually do maybe one slide on binary fission. We say it's really easy. You go from 1 to 2, you just get longer, make a little septum and divide. And then we go into this long, detailed version of mitosis and make you think the bacteria are simple little creatures. I know, I do the same thing with my 2107 students. But in reality, there is a lot more going on. We're going to cover some of it, right? And I want to remind you that when we talk about growth with microbes, we're not talking size, we're talking numbers of cells. So we mentioned that earlier. And there's another definition here, and it's known as the generation time or the doubling time. So literally, it's the time that it takes for one organism to go to 2. And this varies. So under perfect conditions, a lab strain E. coli, one that you buy and replicate in the lab, can double in 20 minutes. So before you leave at night, maybe 5:30, 6 o'clock, you set up a culture, you come in at 8 o'clock in the morning, it is fully grown up and ready to go. E. coli out in the soil where it's colder, nutrient levels are different, might take a lot longer to double, hours. There are some microbes, I had friends working with them in grad school. I was working with microbes, it took about 3-4 days for them to grow up. They were working on ones that would take months. Anaerobic organisms can take 3 to 4 months to get a full culture to grow. So basically you start cultures, and then you do something else for a few months while you wait, literally. Does that make sense? So there's a lot of variability there. Okay, so the first thing to talk about is a set of structures that would combine to form what's known as the divisome. And the divisome is literally what's pictured here. This is how the cell is going to divide. And so there are several different proteins. The key proteins are the FITS proteins, and these are going to interact all around the cell division plane, and you can see there are several different FITS proteins. There's an anchor to hold the whole divisome. So the ZIP-A is literally a protein that anchors. It's anchoring this FITS-Z ring, which is very similar to the tubulin you would see in a eukaryotic cell, to the cell membrane. So this is tubulin-like. We also have an actin-like FITS-A. And so what that's going to do is it's going to help connect the ring and recruit these other divisome proteins. Now the other divisome proteins have different jobs. One of them in particular is known as FITS-I. And this is going to form all around the cell for rods, or only in the very growing center for cocci. So it's helping to figure out the plane for division. Are you dividing long or short? It's called a penicillin binding protein. Do you think our cells want to bind penicillin?
Nope.
So why is it called penicillin binding? Hmm? It's because we're human and we name it by what we observe.
It binds the penicillin.
Oh, okay. Now, the actual septum where they're going to divide is where you have increasing amount of cell membrane and cell wall material being made so that you can elongate the cell and separate them. The other really key proteins are the min proteins. And so these are kind of cute. I like min proteins. These are helping to guide FIT-Z to the actual midpoint of where you're going to be dividing from. So min, C, and D form this spiraling structure. Have you guys watched cement truck? You know how a cement truck will keep circling? Why is it doing that?
Keep it from hardening.
To keep it from drying and hardening. Anybody ever watch the Mythbusters episodes where they try to blow up the interior of the cement trucks to release the cement? This is just me. Okay, never mind.
I missed that.
I definitely
I do watch
Mythbusters,
stuff.
but-
Elaborate.
Go watch. It's not as good as the hot water heater, but it's good. Trust me. Big boom. It's kind of funny. So in this case, they're going to keep oscillating as long as they can. I'm going to switch arms. Might as well get a workout, right? So they're going to keep oscillating. What that's going to do is as they're oscillating, the same way that we can't solidify our cement, we can't divide the cell. Eventually, another protein, min E, will come and it will sweep min and C and D to the sides. And if there's more min E functioning, there's more C and D being swept to the sides so that FIT-C can act and divide the cells. Does that make sense? Okay. So C and D are oscillating. E will push them apart. Once that happens, here's my divisome complex and my FIT-Z ring. I'm going to divide my cell here. And I literally divide by bringing over and making more cell membrane and cell wall.
What happens, so how do you go from the step right above it? Because I see the chain getting bigger and then it just stops.
Where?
Like the red in the middle.
The red is thickening, right? So that's increasing the amount of min E that's going to allow FIT-Z to form there. Does that make sense?
So it
Yep.
forms from many.
We have questions.
Yeah, you have a question coming up. It doesn't form, it recruits it.
Okay.
Yes, you have a question. So to initiate division of bacillus-shaped cells, what ring forms in the center to define the cell division plane? Low-hanging fruit on this one. Low-hanging fruit. Yes, ma'am. Come at the break and I'll do it. Use your eye clicker and then come at the break. And congratulations. I can't say.
Yes. Why do you keep dying on me? We used to be friends. It says the battery's now up. Yeah.
Oh, you're on the board?
Two.
Oh.
Did you?
Mm-hmm. No, because we had extras add in. Oh yeah, no, we only had 2, so everyone's answered? Okay. The low-hanging fruit works, right? Look at that. That's a pretty thing. Let's go ahead and take our break because after this we've got a discussion and another question. I don't want to stop in the middle. Hold on 1 second. Come on. There you go. Yeah. And I didn't have that cute little, like, I needed a wheelbarrow. It was twins. They came a month early. They were still 5 pounds each, but, you know, whatever.
Big twins.
Big twins. It's fun. They're really cute now, though, so it's okay.
Are they identical?
We're 99% sure. That's another story. Anyway, now you know what you want to talk about during lunch tomorrow, right? Yeah. So, well, it's not lunch in here. It's like our 3 o'clock. Please don't fall asleep on me. Snack. So the one thing to talk about is when we think about bacteria, most people think about the fact that they're just going to duplicate really fast. And you're taught early on that they're going to form a single replication fork. And that was the original idea, and that means that they would only be replicating in one direction. They would start in one place and go. And what we now know is a lot of them will do what are known as bidirectional replication forks. They're going to go around the circle. If they have linear chromosomes, they do make multiple replication forks, and that's going to speed up cell division. Okay? So one more really important thing to talk about is MREB. MREB, depending on how it's arranged around the cell, will change the shape of the cell. So what are some shapes of bacteria? Cocci, round. Rod, bacilli. Spirochete or spiral, spirally. So depending on how MREB is distributed, that will greatly affect the shape of the cell. And that's because it's going to help recruit cell wall synthesizing proteins to very specific regions, and it aids with cytoskeleton formation. If you knock out, meaning I eliminate the function of MREB in a mutant, the default is cocci. They will always be round. Even if they are bacillus normally, if you knock out MREB, the default is going to be cocci. Now this is not the only shape regulator. Different types of cells, especially the ones that have weird shapes to them, are going to have different regulators. So Caulibacter has crescentin. Helicobacter has something that's more closely related to crescentin than MREB. And so the point of noticing that is that there are different shape regulators for different classes of organisms. Okay. So we've mentioned peptidoglycan. Where do we find peptidoglycan in the cell?
Cell wall.
Cell wall, yay! Who has more peptidoglycan? Gram-positive. Awesome. And we talked about NAG and NAM, and G stands for NAG and M stands for NAM. And so what we have to talk about now is how we make peptidoglycan. And this looks horrible. It looks really confusing. There are lots of errors and letters, and we're going to break it down and make it a lot easier. But this is epically important because cell walls are important. Those are key targets for antibiotics. That's key for the cell to be able to divide. It's how we're going to identify ourselves. So cell walls are really important, so we do need to know this. So the first thing to consider is you have this thick cell wall. It's really hard to add something that's thick. So what we're going to do is we have to make a little hole, a little opening, in order to build more cell wall. Does that make sense? So I'm going to use something known as an autolysin. And the autolysins are literally going to make a little hole in the cell wall that's there. So it makes a gap in the existing NAG and NAM, and that's going to give me room to fill in the cell wall. I have to get cell wall material from inside the cell across the membrane to this hole in the cell wall. So I need a carrier. I need something to help shift it up. So we have something known as bactoprenol. That's this red thing. Bactoprenol is a very cool molecule. It has a hydrophobic alcohol, and that's going to bind to the NAG-NAM precursor inside the cell. So that's here, okay? It's going to then transport across the membrane. It's bactoprenol, and it's hydrophobic. So it's going to very easily cross the cell membrane. When it does that, there are transglycosylases, and these are the enzymes that are actually going to join these together. They're in the periplasm. They're going to insert this cell wall precursor into the hole, and help make the glycosidic bonds to join them together. So it's make a hole, transport from inside the cell to the hole, glue the hole together with the new material. Does that help break it down? Okay, I mean, you have to know the steps, but the overall idea, open it up, add something new, glue it together. Okay? And so this process is known as transpeptidation at the end, because remember, depending on whether I'm a gram-positive or a gram-negative cell, I'm going to transpeptidate. I'm going to join and cross-link with the amino acids. And so as a reminder, these might be with muramic acid residues in gram-positives, or between DAP and alanine and gram-negative. That should be a reminder for when we initially did the cell wall. I know it was so long ago yesterday, but it was there, right? I promise it's on the slides. Just a couple more. This is an intermediate question because it's asking a very straightforward question, but not in a straightforward way. Trying to give you different types of questions. Yes.
I was just wondering, is this how the wording on the multiple choice?
This is wording actually mostly from your textbook. I use their I think questions, and I don't always use their questions. So not necessarily. That I don't use theirs? Well, I give you a mix. Some of these I've written and some are from the textbook. All right, there we go. And we have 100% getting it correct. So you had to kind of like sort through, what is this beta 1,4 glycosidic? If you think back, we did talk about beta 1,2, 1,4 a little bit. And I said it wasn't that important, and it isn't, because I just want to know where I make my hole and how I make my hole. Right? Okay. And then we get to growth curves. These are excellent for information, and they are horrible to make. So if you work in a micro lab and you discover a brand new bacteria, you're all excited, right? Like you think you're going to name it after yourself, and you've got something. And then your boss tells you, now we need to figure out how it grows. And the first time this happens, you have no idea what's coming. So you're like, sure, I'm ready. And then they tell you to get a sleeping bag. Why? Why do I need a sleeping bag? And here's why. Over a period of, well, it depends on how fast your cell grows, you take measurements to see how fast it's growing. So if you have something that grows over a span of 3 months, like my friends in the anaerobe lab, once a day for 3 months, you take an optical density reading to see how many cells are in that culture. Make sense? And so then what I would do is that's measuring what's known as turbidity. And so I would plot this line over time. If you have something that grows like E. coli every 20 minutes, guess how many times you are taking?
Every 20 minutes.
Every 20 minutes. Hence the sleeping bag.
So sleep is not happening.
Yeah, no. So generally speaking, what you do is you make friends with somebody else and you're up for 4 hours. You go to bed, they're up for 4 hours. And you switch on and off until you get your growth curve done. All right? And this will change in different conditions, but it says in a given condition, under certain growth, nutrients, temperature, what have you, this is how the cell grows. And so cells will always grow in a set way. They have a lag period. You put them into culture and they freak out for a moment. What sugar source is here? What protein source? Do I have nitrogen? Do I have to fix my own? What am I doing? The lag is literally where they are trying to figure out how best to use that culture media. They're going to turn on enzymes. They're going to start metabolic processes. but they're not growing because they're not doing it yet. But once they get up and running, they go into what's known as the log or the exponential phase. This is the fastest rate of growth. Life is great. There is a whole smorgasbord of food. They're going to eat all of it. They already have their enzymes ready. They're going to keep growing as fast as they can. What's going to happen over time, though? It's going to get overly crowded, and they're going to eat all the nutrients. And, well, sometimes. And they're releasing waste products, so they're polluting their own environment. So there's a lot of bad stuff happening. And that's where we're going to hit the stationary phase. They're starting to use up their ingredients. We're starting to see some cells dying off. We do have new cells growing, but we also have cells dying. And then eventually, the overall cell population is going to die. Notice this number still stays pretty high. Dead cells still make it cloudy in the media. It still keeps a high optical density. But the red line is measuring what's known as the viable count. If I were to transfer these cells to fresh media, how many would be alive that I'm transferring over? Does that make sense? And so this is really important for a few reasons. Depending on where you are in the growth curve, your cells are doing different activities. So let's say you're studying biodegradation. They might not be very active here. They might not be very active here. They might do their most biodegradation here. Or they might only do it at the very, very beginning. And so by doing this over time and activity, you might know, okay, if I start a culture at 8 a.m., I have to pull my sample at 2 p.m. That's when they are the most active. Somebody else in your lab might not be using cells for that enzyme. They might be looking at something else, and they might need to pull it at 4 o'clock, not 2 o'clock. So it's a way to gauge activity, and it's a way to gauge how lively your cell culture will be. But again, it's going to shift depending on conditions. Does that make sense? Let's see. This is a hard question, in my opinion. So take your time.
What's LB?
LB is a complex media. It actually stands for Lariobroth. Doesn't matter in this case. Complex, yeast, tryptone, peptone, all of that. This is one of the harder questions in this unit, in my opinion.
But it's in a new place, so it's got to figure out what kind of that. Sure, it's
Two more. So far, you're doing well. Not 100%, but pretty well.
That's why it has a long black face, right?
I'm stopping right now. Don't talk anymore. Don't change anything. There you go. Good job. Well, I was afraid if I started talking, somebody would change their answer, and I didn't want to do that. So in this case, you guys got it. That's awesome. Just because I'm going from minimal to rich doesn't mean that it's easy. How many of you have ever gone home after a semester living on like minimal food and get sick because you're eating all that rich food? Or you're like me, where during the semester you're not cooking as much, and when you have a break, you all of a sudden start cooking in mass. Yeah. Oh no, I make.
Or like me, cooking every day and don't have time to study.
That. See, if I did that, I'd be happier here.
I know,
Yeah,
but see, you cannot
Doesn't work
win.
very well.
You cannot win.
No, you probably can't win. But the nutrient-rich food can sometimes take a lot out of you, right?
Interstitial stomach.
you still have a long lag phase. You still have to get used to the food. Does that make sense? When I was in college, you know, you go through like lean phases. I had a rice cooker. I would buy the cheapest American cheese slices I could and rice vinegar because it was cheap. Oh, don't even give me that look. You either have noodles or rice. That's cheap. You throw it in, you've got your protein. No?
American cheese and rice wine vinegar and rice.
Mm-hmm.
Oh
Sounds
my gosh.
disgusting.
Thanks, guys. Did it taste good?
No
Nope.
Was this with your aerobic fitness instructor, your roommate?
Yes, she was living there. She didn't eat that food. No, she ate the 49-cent Tostino's pizza things, but those.
They're going up to
Okay, but those are like 40
$1.25
grams
now.
of fat. I was trying to be healthier.
So you replace carbs with fat?
You guys are soldiers.
Didn't work very well.
I had to get through school. I was cleaning rat cages to pay my way through. It was not pretty, but we did it.
Smells great.
Yeah, you have no idea. Try the Vols, those are worse. Anyway,
Try the one.
so sometime, what we've been talking about so far are the voles. They're a type of rodent, nasty little thing. Don't like them. These are what we've been talking about growing. So these are considered batch cultures. This media would have been clear. I put an inoculum in. I let them grow overnight, and I get a nice thick turbid culture. But by the time I'm done with this culture, it's dead, right? Remember, it's at the end of the stationary phase. So what did we say was happening during the growth curve that took us from the log phase to the stationary? My nutrients are running out,
Build up with back.
buildup of waste, and too many cells. So sometimes you get hired by a company or the government, and you need to have a high number of cells. And you need them at a very specific spot in that growth curve because I want them, let's say, biodegrading something. Does that make sense? So I don't want to use batch cultures because I can't get them all doing the same thing at the same time. It's pain. So what you use instead is something known as a chemostat. In a chemostat, it's a closed system, but you have these ways, these valves, where you can put in fresh media and or pull off waste and dead cells. And so what you can do is build up a culture and a chemostat to an extremely high density and then go take it somewhere. So in our case, we didn't have fancy glass chemostats. We actually used empty pony kegs. And we would take these pony kegs filled with bacteria to government facilities and put them in the ground to help degrade the waste. So all it is a closed vessel where you can regulate the flow in of fresh media and food and the outflow of waste and dead cells.
So is that, they make this fish tank that as you put water in, it pours it out essentially? Is that kind of what it's doing with that?
I don't know anything about that tank, so I can't say for sure.
Yeah, because it's like keeping it, as you replenish nutrients and stuff like that, and like it pushes everything out into it.
Pushing everything.
Like the waste.
The concept is the same, yes. I haven't looked up that tank, so I don't know. But the concept is the same.
I was just gonna ask if you could explain how it works.
So it depends on what you're trying to do. Literally, it's like one of these, except that I can say, oh, I have a lot of waste coming out. I want to pull some of that, but I'm going to replace it with fresh media. It's like washing out the waste and adding in the fresh. Does that make sense?
Yeah.
If you're cooking a pot of pasta, it would be like saying, I don't want to waste my water. So as my pasta, this batch of pasta is soft and ready, it's like al dente, I like it. I'm going to pull that out. I'm going to keep my water boiling though, and I'm going to add in fresh pasta. Same concept. We're on a whole eating thing today, right? It's been a few hours since I've eaten. I'm hungry now. Okay. So now we move towards measurement. We talked about turbidity that measures the total number of cells, but also your material that you're growing them in. If you're growing them in oil or something, that's also cloudy. So there are a lot of problems with measuring. One thing that you can easily do is you can try to count them. You can't do this with the naked eye unless it's Theo Margarita. You're going to do microscopic cell counts. If you've ever tried to stare under a microscope for a long time, your eyes start to tear up, blur. You can't tell what's an eyelash, what's a crack in the cell, and what's something real, right? So what we have are these gridded cells, and you put a very specific amount of cell culture on it. So maybe you do a half of a mil. And you look and you count per quad how many cells are present. And then you extrapolate that to your overall culture. Does that make sense? So if I know in a half of a mil, I have 1,000 cells, then per mil I have 2,000 cells. And then if I have 10 mils, I have 20,000, right? Did I do that math
So it's
correctly?
kind of like a statistic population.
Kind of. But here's the problem. Are those alive or dead?
They're alive.
I have one alive, one dead, and everybody else sitting there like, no idea. How would I know? The rest of you who are being quiet are correct. I have no idea. I don't know if those are alive or dead. Okay? Although I really appreciate those who are trying. So I don't know if those are alive or dead. If you think back to when we did fluorescent microscopy, we talked about DAPI staining, where we looked at the nuclear information. Again, I don't know if you're alive or dead. I just know that you're a cell. And sometimes that's okay, and sometimes that's not. But sometimes I want to know my viable number. I want to know how many are actually alive and able to grow. So in this case, you could do a viable cell count. A viable cell count can be done in two different ways, depending on whether you're looking for aerobic organisms or anaerobic organisms. But the overall idea is whether you're putting the agar on top or the microbes on top, I'm going to take a set amount, again, another maybe 100 microliters this time. It depends on the cell. I'm going to put 100 microliters of my cloudy culture, so like something from here, and I'm going to put them onto my plate. And instead of streaking it like we looked at, I'm going to use what's known as a hockey stick or a Bacti spreader, and I'm going to try to spread it all over that agar. So I want to try to make one smooth amount of all of this media. Follow me so far. When they grow up, I'm going to get all of these little dots on there. And so it'll be on top if they're aerobic organisms. I might have subsurface colonies if they're anaerobic organisms, so you can alter this. But then you would literally stand there. You have certain types of equipment to mark off the colonies that you've counted, and you're trying to count the colonies all over the plate. What do you think TMTC means? We use that a lot in lab notebooks. Too many to count, exactly. So I will tell you from personal experience, once you get over about 300 or so of these colonies, your eyes are blurring, they're tearing up, and it hurts. So you have a way to say TMTC. I'm done with this. How can I avoid that? How can I get an accurate count and not have too many to count? Any ideas?
Grid.
I can do a grid.
Dilution.
Serial dilutions. Remember those painful things from chemistry? They actually serve a really good purpose though. Instead of 100 microliters, if I get too many to count, and this is too many to count, this would hurt. This is verging on that probably like 1,500 or so. That's hurting. You're going to do 1 in 10 dilutions. And so here's my sample that I'm counting. I do a 1 in 10. I kind of mix that up, 1 in 10, and so on and so forth. And then I'm going to plate them. Too many to count, too many to count. Here, if you stand and count, you get, in this case, they said 159 colonies. Okay? A 1 in 10 dilution gives me 17 colonies. Is that about accurate? Yeah, because this a true 1 in 10 would be 15.9. So that's pretty close. And then I do another 1 in 10 and I get 2 colonies, and that's close to a 1 in 10 dilution again. And so if these all make sense, you would then multiply back out by your plate dilution factor and get an idea of your, not cells, but colony forming units. I don't know how many cells are in here, but you'll see reports where they say CFUs, colony forming units, because remember each colony is a single type of unit of cell that will grow up over time. Okay? So this is a problem because certain types of media can be selective and select against certain organisms. They don't all grow in the lab, so this is not perfect, but it's a good way to get a viable cell count. And this would eliminate the concept of all cells. I know that these are all viable. It's not the same as looking at turbidity where they could be living or dead. Does that make sense? Okay. This is another kind of hard, kind of not.
I mean, you could also have errors between dilution.
Like
You have
it
dilution.
decreases it.
But if
So
you
it
don't
depends on
dilute
the
properly,
number.
you're not basically gonna be.
So if it becomes properly and accurately, it does decrease the error. Because who's gonna sit there and count like 100,000? So these are for sure.
Bees for sure, right? I'm debating.
B is right. Like B is right. For sure.
But see, same thing.
Because it doesn't eliminate errors, but you still have that,
But it
because it definitely decreases it.
yeah,
Decreases
because you
your
still
error.
might have human here. You're still
Because
human.
you can always miscount,
Yeah.
or not count, or skip count.
Or
But it's not saying it's
causing
eliminating, it's just saying it decreases.
Because those are your 159 to 17, which is pretty close.
All right, I've got 12 answers. I'm not loving your responses.
The majority? Or just a couple?
Majority.
You
can use
Come
these.
on.
Did everyone say D though?
Did everyone say
I said D.
I said B.
I said D.
I said D.
The majority is right.
D is wrong.
So, I mean,
The
I don't think
majority
it
is
decreases,
wrong.
the dilution doesn't decrease the.
So the
error.
majority said,
so that means B.
It could decrease it.
It does decrease it.
Yes.
We just talked about it.
So I'm not going to count it wrong on this, but I want to have a discussion about this. You are right that it helps to figure out mathematical issues if you do it correct. How many times do you think dilution error, serial dilutions work perfectly because of human error, though?
Very rarely.
Very rarely.
Yeah.
So it would increase error every time you're going in. Serial dilutions are only done to reduce that too many to count issue. Does that make sense? Okay?
There's some human errors you can screw up, but it doesn't decrease any error.
Right. Okay. So just one final thing about how you would measure. For spectrophotometry, this is where you're measuring cloudiness. That's the optical density that I mentioned, that turbidity. But the one thing that I wanted to mention, and I said it already, I just don't have a slide for it. You have to be very careful because other things can make it cloudy. Oil can make a media cloudy. Excess sugars, excess proteins can change the optical density. And so when you are measuring this, you don't measure against water. Your blank that you're comparing to always has to be the media that you're growing them in. Does that make sense? Okay. So we're going to switch to how we would encourage growth and how we would stop growth. There are cardinal temperatures and cardinal pHs and cardinal oxygen levels for microbial growth. And what that literally translates to is that an organism is best fit for its environment. Its enzymes prefer that environment. The cell membrane, the cell wall, all prefer that environment. So remember we talked about that archaea have different types of cell walls depending on their environment, different types of membranes. It all goes together that they have kind of their niche that they like and they're adapted to it. And one thing to consider are enzymes. Enzymes have an optimal temperature that they work at. So proteins where it's the optimal. If you go too far below, they cease working. Too high above, they denature. So this optimal or optimum temperature is where they like to work. And very often that'll be the niche that the organism is living in. So microbes have different temperature classes, and there's a wide variety amongst them. And this is all prokaryotes now. The ones that we hear most about are the mesophils. These include things in your human microbiome like E. coli, and it includes pathogens like E. coli. So anything that can live in you is a mesophile. There are organisms, like I said when I mentioned Lake Vostok, that will live and act inside of an ice core. They like it. These are our psychrophiles. You guys refrigerate foods? I know, psycho for psychrophile, that's where I'm at, it's okay. You refrigerate. If you ever put your milk in the refrigerator and totally forgotten about it,
Yep.
and you pull it out like a month later and it's yellow liquid at the top and cheese on the bottom? No, just
Yep.
me?
No,
Okay,
that's-
yep, thank you. Was something living and growing in that milk?
Yes.
Yes. Refrigeration helps slow growth, but there are things that can still grow in it
That's why
happily.
it's stress.
Exactly. Now, on the other hand, we have thermophiles. Now, something like Geobacillus that's a thermophile that likes to grow at about 60 degrees is never going to impact you. You're only about 32 degrees. They don't like that. You're too cold for them. But they like it warm. Then there are hypothermophiles that like it hotter, 85 degrees. We have extreme hyperthermophiles. Paralobus is one of our hottest bacteria, likes about 106 degrees. There's another line here of archaea. They found an organism that likes to be at about 120 degrees. So autoclaving, which is one way that we kill them, I'll talk about that in a few moments, doesn't kill them, they like to grow in there. So each organism has different temperature classes at which they grow. And they have adaptations to help with that. So for instance, if you look at a psychrophile, they have many more alpha helices and decreased beta sheets in their proteins. Their cell membranes have more unsaturated and shorter chain fatty acids. They have a lot of exopolysaccharides on the outside to coat and protect them. And they have a high percentage of cold shock proteins, cryoprotectant, and no joke, antifreeze proteins to help prevent gelling up of the cell. On the other hand, thermophiles and hypothermophiles have heat-stable enzymes. They increase the number of ionic bonds between their charged amino acids in their proteins, and they do produce solutes to help them resist denaturing and unfolding of their proteins. They have more heat shock proteins. So these organisms are really well adapted to the different temperature classes. pH is another one that we will talk about extensively. Do you remember when we looked at that redox tower? I know you're trying to blank it out, but remember we said oxygen and then nitrate? I said I'm going to skip this iron because it's pH 2, this doesn't really happen. Okay, hold that thought. There are organisms that are acidophiles. They need it to be below 5.5 in order to grow. One of them that we will talk about a lot later in the semester, like a whole few days from now, Acidothiobacillus peroxidans will actually acidify its environment so that it's able to metabolize iron faster. So it will drop the pH to a maximum, of 2. The chemists have figured out a way to detect pH levels below 0. I still have not figured this out. My bio brain has difficulty assimilating that, but they calculate it. It's math, so I'm going to stick with my bio. But Acidothiobacillus can survive at extremely, extremely low pHs. If you are talking about an acidophile, they have a lot of increased tolerance and stability in their cell membrane. They actually will use proton concentration changes. So they'll use a sodium motive force rather than a proton, a hydrogen motive force. And if you alter the proton concentration to make it more neutral, you actually will pop open and lyse the cell membrane. They cannot tolerate neutral pH. On the other hand, we have alkylophiles. These grow best at a pH of 8 or above. Things like soda lakes, which are about 12 on the pH scale, they like it there. They need that pH. High carbonate soils. You see a lot of our halophilic or salt-loving, desert-loving organisms will also be alkylophiles, and these will also use that sodium motive force because they can't use the proton motive force if they are dealing with pH changes. Does that make sense? Anything that's going to live with you is typically going to be a neutrophile. Again, that's where our E. coli will fit in. I mentioned halophiles. These are our salt or low water content living organisms. We have our halotolerant organisms, and some of our pathogens are considered halotolerant. E. coli and the others are considered non-halophilic. You just can't mess with them. They need water. You go from halotolerant to halophiles. Alivibrio fisheri, we're going to talk about, they live in salt water. So they like ocean life. You also have extreme halophiles. These are things that you would see in things like the Dead Sea or in the desert. I mean, they like it that dry. And so the water availability expresses water activity. How much water do they need? So for example, human blood and pure water, this is our normal non-halophilic organisms. And then it starts to go beyond that. So we have seawater, bread, maple syrup, and ham. You usually think of maple syrup and salty ham as protecting against growth, but Staphylococcus loves that. They're totally cool with that. Again, our processed meats, salami, Saccharomyces yeast, love them. Sugar in jams, salted fish, dried fruit. You're starting to continue to see organisms that grow in it. It's a smaller number, but some of them preferentially like those preservatives that lower the water availability. Does that make sense? Okay. So then we move into oxygen. So I'm just kind of giving you the highlights, the examples of the different things that would enable them to grow, and then we'll switch to kill them for the rest of the day today. So oxygen requirements, if I'm looking at organisms growing in tubes, if they're growing at the very top of the tube only, these are aerobic organisms, all right? So air is about 20% oxygen. They're going to grow towards the air. As our oxygen level depletes, they can't grow. These guys are a little bit different. These preferentially grow at the top. They can continue to grow. You imagine that my oxygen level is going to maybe start here, and this is where they're going to stop, because these are obligate anaerobes, like Clostridium. They cannot grow in the presence of oxygen. It will kill them. These guys are just growing everywhere. And the trick is they're facultative. They grow heavier where there's oxygen because you can get more energy if oxygen is your terminal electron acceptor. But they can grow throughout. They would just use different electron acceptors. These guys are microaerophilic. They like oxygen, just not too much. Too much will kill them. It's like too rich of a food. There's no difference here in growth patterns. These are anaerobes, but they're aerotolerant. They can live up here. They can live anywhere. They're not using the oxygen, so it doesn't impact them. Does that make sense? Okay. And so now, like I said, we have to figure out how to kill them or how to grow them with those different classes. So I can alter my oxygen concentration, and let's say I want to kill the anaerobes, I put them in a hyperbaric chamber. Or if I want to eliminate eptom, I put them in a vacuum so they can't grow. If I have something where it needs to be anaerobic, you can put them in an anaerobic chamber. There are specialized gas packs which will eat up all of the oxygen, so there's no more available in here. I've even had students, when this would run a little expensive, we would use mason jars. We would put a tea light candle on top of the stack of plates, light the tea light candle, and seal the jar. As the fire consumed all the oxygen and then burned out, you've used up all the oxygen in the jar, and now you can grow your anaerobes. Does that make sense? So these are just ways that you would grow them depending on what you need. But oxygen is not always great, which is why I said we're going to grow them and then we're going to kill them. When you've gotten a cut, anybody ever put hydrogen peroxide on a cut? What do you see?
Bubbles.
Bubbles. That's like one of the greatest things. Kids freak out if you touch a cut. They don't want you to touch a cut. Like, wait, look, science! And you pour hydrogen peroxide on it and it bubbles. Sometimes they're still pissed at you because it burns a little bit, but you try. But you're seeing these bubbles, right? What you are testing here is for the presence of catalase. If you are an organism that does not have that, you can actually destroy that. But you have some catalase present. And when you have catalase present, you're going to take hydrogen peroxide and convert it into water and oxygen. And so you're seeing these bubbles forming up. So oxygen can be rough. Superoxide, hydrogen peroxide, hydroxyl radicals can all damage cells, both microbial cells and your cells, because they're really just targeting the cell membrane. Okay? So in the same way that you might eat something that has, quote, antioxidants in it to protect yourself, cells have enzymes to protect themselves. So if you shoot out peroxides or superoxides as part of your immune system, which you do, they can defeat against it. Catalase breaks down hydrogen peroxide into water and oxygen, makes it a completely safe molecule for the cells. Peroxidase is going to break down hydrogen peroxide also to water, but it's going to consume energy to do so. It's going to use NADH. Superoxide dismutase is going to specifically work on superoxides, but what does it generate? Hydrogen peroxide. So if you're going to use superoxide dismutase, you need to use it in combination with catalase, because otherwise the hydrogen peroxide can kill the cell. There's also superoxide reductase, which is going to work in a similar manner. It's going to use a different reducing agent, but it's also going to generate hydrogen peroxide, so you would want to pair it with catalase. Does that make sense? So if I want to control microbial growth, I have a few terms for how I would do it. If you hear that something has been decontaminated, that means it's safe for you to touch and to handle it. So in other words, if there's been a sick patient in a room and the room has now been decontaminated, it's safe for the next patient to go into the room. If you see that something's been disinfected, you are specifically targeting pathogens. The quote good bacteria are still there. But disinfection, if you look at Lysol, it doesn't say it kills everything. It says it kills the harmful pathogens in this list. It's disinfecting, it's targeting just those cells. And so there's a difference in those definitions. Overall, what you're looking for is a way to decrease the potential pathogenic load, or in some cases, all of the microbial population. One key way to do this is with using heat. So heat sterilization, you get what's known as the thermal death time. How long do you have to hold the cells at that temperature to kill off the pathogens and or all of them, depending on your goal? So you have a couple of ways you could do this. Autoclaves, as I mentioned earlier, go to a very high heat, about 120 degrees. They also do pressure. And so this is pressurized steam, which means you can typically put them at that temperature for a lower amount of time and have an effective kill. There's not a set timing for autoclaving. It depends on how much you're doing. Smaller amounts of fluid, you would autoclave for a shorter amount of time. Larger volumes, if you're autoclaving liters of culture, you're going to have to put them in for much longer. Does that make sense? Okay. You mentioned pasteurization. Pasteurization uses heat, different types of heat, for different amounts of time to try and target the pathogens. All it's really doing is lowering the number of microbes present. Over time, they can build up again and turn your milk into cheese. But it takes a little bit, so if you consume the milk in that period of time, those microbes are not going to harm you, and it's not going to affect the overall milk until their numbers get so high that they start metabolizing it. Does that make sense? One other thing that I can show you is radiation. If you work in a lab, very often what we will do is use radiation overnight. And so UV radiation will damage you, damages your cells. It damages the DNA, causing them to die. The other form of radiation that you can use is ionizing, and that's going to generate free radicals, which will then go and target macromolecules, typically DNA and protein, destroying those and killing the cells. The different cells have different ways of surviving this. If you're a spore, you're pretty good. We can bombard you 3,300 grays of radiation, and you're fine. That's a tremendous amount of radiation. Viruses need a lot of radiation because they're not alive, and they have these really great protective measures. But if you notice, Salmonella only needs about 200 grays. So it depends on what they are. The best surviving, living, viable vegetative organism is Deinococcus radiodurans. And these can survive over 2,000 grays of radiation because they can repair their DNA as it's being damaged. I'll leave you with that thought. We're going to finish this chapter tomorrow. Awesome? Cool. Thanks, guys.
PowerPoint, you know that this is 1 through 9, and that's what the test is on. Are we going to finish all of
I'm going to try my best. If we don't finish, I do not test on things that I don't cover. I would shift it to exam 2. Does that make sense? I will never test you on material that we do not go over. Awesome. Thank you.
Was there any other handout in the next Tuesday?
No, I know you weren't here. No, all the notes are online, but you did miss. Yeah. Did you pull the syllabus and the notes yet? Okay. So I would choose